A very good morning, good afternoon, and good evening to all the participants and colleagues in different time zones. My name is Sridhar Bhavani, and I head the ROS research here at Summit. And before I move on with my presentation, I'd like to thank the organizers of the technical workshop for inviting me to present today. And the title of my talk is A Decade of STEM-RAS Phenotyping Network that I coordinated and collaborated over the last decade in East Africa, trying to reflect on the opportunities, challenges, and the way forward. Just giving you a historical perspective as to why UG99 was considered as a global threat. More than 80% of the wheat varieties grown worldwide were found to be susceptible when tested in Kenya as early as 2006, indicating the global vulnerability of wheat germplasm to this devastating disease. At the same time, it didn't remain confined to Uganda. It spread out to 13 different countries, all the way from South Africa to Middle East, all the way up to Iran, as you could see in the slide. And this race was not only unique, highly aggressive with broad virulence spectrum, defeating over 50% of the known STEM-RAS genes, including the widely deployed STEM-RAS gene SR31, which was quite effective in controlling STEM-RAS for over 3 decades. And interestingly, it didn't remain the same. 14 races within the UG99 lineage have been identified, which have overcome several race-specific genes, and of which 7 races have evolved in Kenya, suggesting the hotspot for the evolution of the UG99 race group. And significant risk would have been if this race migrated into other regions of Southeast Asia, Central Asia, and the Pacific, wherein close to about 35 million hectares of wheat would have been affected and would have been vulnerable to stem rust, indirectly affecting the lives of about 1 billion people in those wheat growing environments. And knowing of the fact that stem rust can cause significant epidemics with susceptible varieties, yield loss predictions were close to about 70% if this disease migrated out of East Africa into other wheat growing regions. And in order to mitigate the threat of this disease, the Borlaug's Global Rust Initiative was formed, largely with the leadership of Dr. Norman Borlaug, sounding the alarm as early as 2005. The Rust Summit was held in Kenya in 2006, and BGRI was officially launched. Multiple projects were funded in the last decade, with the durable rust resistance in wheat in 2 phases, followed by the delivering genetic gains in wheat, and very recently, the accelerating genetic gains in wheat. And all of these projects have several aspects of research on mitigating the threat of UG99 and other transboundary diseases. And BGRI reflects a true power of international cooperation and collaboration of different research institutes, international as well as advanced research institutes and national programs, all working under one roof, trying to mitigate the threat of this disease. So a part of this DRRW project was to establish 2 phenotyping platforms in East Africa, one in Kenya and one in Ethiopia. As you can see, Kenya represented a hotspot environment for testing against the UG99 race group. Similarly, the platform in Debrasite represented a phenotyping platform to test against the Durham races, which are quite unique to those environments, alongside UG99 as well as the Diggle race groups. Both phenotyping platforms facilitated 2 seasons of testing, and at the same time, ease of germplasm exchange of introducing international germplasm for testing. Something very important with Kenya was to extend the shuttle breeding all the way from Mexico, and majority of the facilities were supported by BGRI, and also the fact that we had expert scientific capacity on the ground operating these 2 phenotyping platforms. These 2 phenotyping platforms form a part of the International Wheat Phenotyping Network that spreads across different regions of the world, across different traits, such as yield potential, various biotic and abiotic stresses that wheat germplasm can be screened across in different parts of the world. And as CIMMYT has a mandate to deliver a combination of traits to its IWIN partners, these phenotyping platforms play a very important role in disseminating not only high-yielding varieties, but a combination of traits, biotic as well as abiotic stresses, which can be released in these target environments as varieties. And something which I wanted to reflect on is these 8 phenotyping hubs that reflect the International Disease Phenotyping Network, which encompass most important diseases such as the 3 Rusts, Septoria, Fusarium, as well as most recently the wheat blast disease. And these phenotyping platforms help in evaluating of traits that can't be screened in Mexican sites. And they are done in conjunction with NAS partners, where environments are optimal for specific trait phenotyping. And these hubs generate high quality phenotypic data under defined agronomic practices, and also represent hotspots for pathogenic diversity, facilitating both evaluation and selection, as in the case what you would observe with the STEM-RAS phenotyping platforms. This is just a snapshot of all the activities within the STEM-RAS phenotyping platform, which I'll be talking to you a little bit in detail. Here is a reflection of the International STEM-RAS screening phenotyping platform established in Kenya. So far, over 600,000 accessions have been screened since 2005, with an annual capacity of 50,000 lines split over 2 seasons from over 20 to 25 countries. And as you can see, the wheat accessions comprise breeders, advanced breeding lines, segregating populations, mapping populations, GWAS panels, and land races that are being evaluated over multiple years. And similarly, in case of the Debrasite phenotyping platform, we've evaluated close to about 150,000 accessions since 2007, with an annual capacity of 10,000 lines. And here's a reflection of the screening of over 15,000 lines from the USDA National Small Grain Collections. As I mentioned, the Durham races are quite unique with combination of virulence to stem rust genes SR13 and SR9E. Something which is quite important in the context of CIMMYT breeding activities was to incorporate resistance to stem rust within the breeding materials that are distributed globally to all the cooperators. So trying to take the opportunity of shuttle breeding that was designed by Dr. Borlaug as early as 1940s, using two contrasting environments in Ciudad Obregon and in Toluca, basically advancing 2 generations in a year, trying to cut the breeding cycle by half, but also at varying day length regimes and at the same time altitudes and different elevations with a combination of different biotic and abiotic stresses, trying to make the germplasm not only widely adaptable, but also resistant to biotic and abiotic stresses in other environments. So using this opportunity and to incorporate stem rust resistance, the shuttle breeding was extended all the way to Kenya as early as 2008. And as you can see, close to about 1,500 F3 and F4 segregating populations undergo the shuttle breeding scheme, wherein 2 generations of advancement and selection following the single backcross selected bulk scheme is being implemented. Alongside segregating populations, advanced lines, close to about 80,000 that constitute the stage 1 and about 1,500 that constitute the stage 2 yield trials, are also evaluated every year to ensure that the germplasm that gets distributed as the part of the International Wheat Improvement Network carries a combination of high yield potential as well as stem rust resistance within the breeding materials. And the first products of these shuttle breeding have been delivered worldwide since 2011. And knowing of the fact that UG99 was easily able to overcome several of those race-specific genes, CIMMYT's focus in terms of breeding was to deploy pleiotropic slow-rusting adult plant resistance genes to enhance durability of resistance within the breeding materials. And the hypothesis behind this is a combination of 4 to 5 minor genes, which are in additive interactions, can actually enhance the levels of resistance to almost near immune levels. And some of those pleiotropic genes are quite widely present in CIMMYT germplasm, such as LR34, 46, 67, 68, alongside the stem rust gene SR2. And multiple other QTLs have also been identified within CIMMYT germplasm. So the interactions of these pleiotropic QTLs have actually enhanced the levels of resistance within the CIMMYT breeding materials. However, as you could see in the picture, the individual effects of this minor race non-specific genes is quite low. One such important gene in the context of stem rust resistance has been the SR2 gene, which was identified as early as 1920s, conferring effectiveness over 100 years. And this gene has been widely used by Dr. Borlaug and still forms a backbone of several CIMMYT wheat germplasm. And even though this gene by itself only confers modest levels of resistance against stem rust, combination of SR2 with other minor genes has been shown to work as an SR2 complex in trying to elevate levels of stem rust resistance within the breeding materials. And as you could see, a phenotyping marker called pseudo-black chaff linked to this trait is quite easy to detect within the breeding populations expressed both in the glutes as well as at the internodes of the stems. And significant progress has been made in terms of breeding UG99 resistant varieties, and breeding materials have been released as a part of the international trials in nurseries. And in summary, over 10 to 15% of the lines carry high levels of adult plant resistance, which are usually released in the primary risk environments, such as East Africa. 40 to 50% of the lines carry adequate levels of adult plant resistance, and also combinations of race-specific genes, about 20 to 30%, with good adult plant resistance genes in the background to ensure good gene stewardship. And some of the APR sources that have been developed in CIMMYT are indicated here, alongside some old Kenya varieties that also offer diverse range of options for breeding for stem rust resistance. And this stem rust phenotyping platform is also amenable to yellow rust infections, when conditions are favorable, and also the fact that you have a diverse race group of PSTS11 and PSTS2 race groups that dominate East African region. So evaluating breeding materials as a part of the shuttle breeding, and at the same time with the advanced lines, helps us to be ahead in terms of breeding yellow rust resistant varieties for target environments. As you can see, a larger frequency of lines confer good levels of resistance to yellow rust, not only in Mexico, but also in Kenya, and also in hotspot environments in India. And genetic dissection of some of these genomic regions suggested the involvement of some of these yellow rust genes, such as YR48, YR54, YR60, YR67, and more recently, the YR-Francolin gene. Something which I wanted to compare was to see if there was any effect in incorporating stem rust resistance into high-yielding backgrounds, if it had any impact on grain yield. And comparing the two shuttle schemes, the parallel Mexican shuttle and the Mexico-Kenya shuttle, as you could see, there is a large frequency of lines, more or less in the same frequency that you would observe, both in Mexico shuttle and Kenyan shuttle, and the performance of those lines over standard checks of Borlaug and Kachu. So about 26% of lines performed better over Borlaug through Mexican shuttle, and about 19.5% performed better over Mexican Kenyan shuttle over the standard check Borlaug. So there was no significant difference in terms of the number of lines that could be identified which have combination of high yield and stem rust resistance using the two different shuttle breeding schemes. With this, we were able to actually combine stem rust resistance into high yielding backgrounds. And as I also mentioned, one of the activities within the stem rust phenotyping platform aims at providing reliable phenotyping data for gene discovery and characterization efforts. Over 35 genes and QTLs have been mapped in the last decade, conferring resistance to the stem rust race groups within East Africa. This is a nice little representation by Matras from CDL. As you could see, multiple genes have been published. Similarly, efforts were to characterize new sources of adult plant resistance within CIMID gene germplasm. Multiple biparental populations resulted in the identification of 13 genomic regions, of which some are known to be validated. Similar studies have been conducted, multiple biparental studies, and something which I also wanted to reflect of a recent study that was published in Nature Scientific Reports is the genomic regions associated with stripe rust, both in India as well as in Kenya, trying to reflect the broad effectiveness of some of these genes in different environments. So the 2B region that you identify in black is usually associated with YR31 gene derived from pastor, or YR-Francolin, which is quite widely used nowadays in CIMID. The 2D region, which confers effective resistance in India and Kenya, and also partly in Mexico, is through YR54, moderately effective adult plant resistance genes, quite widely present in CIMID germplasm. The chromosome 4D, that is associated with yellow rust gene YR28. In addition to that, as you can see, the ones highlighted in green represent the 2NS translocation. However, the effectiveness of YR17 is not quite pronounced in environments such as Mexico, Kenya, and Asia, because we have PS2 races, PSTS2 races, which confer virulence for YR17. So there's an additional locus on the 2NS translocation, which also confers resistance to yellow rust. And similar observation has been done in the second stage yield trials of this year, 2018, 2019. Similar trends were observed for yellow rust. However, in case of stem rust, in addition to the pleiotropic APRs, something quite significant was to be associated with stem rust gene SR47, the 7A associated with stem rust gene SR25, the 4A QTL associated with resistance derived from a North Dakota line, and 6B, which happens to be quite unique, which needs further characterization. In addition to that, improved survey and surveillance, trying to monitor the spread of these UG99 races, and also trying to assay for the diversity of these race groups in different regrowing regions of Kenya, trying to collect samples and also to share with global rust reference centers to assay if there is any new race that has evolved in the region. The samples are sent to CDL Minnesota, to Canada, to GRRC Denmark, as well as to Turkey. And this helps in accurate representation of different pathotypes in different regrowing environments, and the information is shared across the global pathotyping lab network. Something which has been significant in terms of mitigating the threat of UG99 has been to develop and deploy UG99 resistant varieties. And Kenya has been the direct beneficiary of receiving both the first and second year yield trials directly from Mexico. And the best performing lines in terms of the agronomic type and stem rust resistance were actually advanced as Kenya stem rust observation nurseries, which form the Kari breeding pipeline, and over 17 varieties have been released in the last decade. These are a reflection of some of the varieties released in the first half, of which some of them have been overcome by the UG99 race group. And the diversity of race-specific genes and APRs in combination reflects the fact that in an event of some of these varieties being defeated, you would have other varieties to easily pick up and replace those older susceptible varieties. Similar studies have been done in Ethiopia looking at the wheat impact studies of CIMET using DNA fingerprinting technology. As you would see, 89% of the samples that were assayed for were CIMET derived. About 55% of the households were growing rust-resistant varieties, and 45% of those samples are related to those varieties released in the last 10 years. And something which is quite important in this context is the spread of an earlier generation adult plant resistant variety that was released in Ethiopia called Kakaba, released in 2010, became quite popular and now currently occupies about 40% of the area. And several other APR varieties have been released, such as Danda, which is also popular with farmers in Ethiopia. Kingbird, which was released in Kenya, has also been released in Ethiopia. And a recent release of Arab Blue, also called as Deka, in 2018, which is an improved version of Danfei both for stem rust and also for yield and yellow rust resistance. Similar impact has also been observed in the USDA breeding programs. Screening was initiated as early as 2005. The only known adult plant resistant varieties were Thatcher and Crisp prior to 2008. And with the screening efforts and the breeding efforts, over 7 varieties with good levels of adult plant resistance have also been released in the US. And this reflects a significant achievement in terms of deploying UG99 resistant varieties in different environments. Over 270 varieties have been released in 11 at-risk countries. And the important take-home message is of the fact that majority of the varieties are released in secondary and tertiary risk environments, even before the UG99 has actually migrated into those environments, and occupying at least 5% of the area. So that is a significant success in terms of dissemination of improved varieties carrying UG99 resistance, and which are a product of testing at the phenotyping platforms in Kenya. Something which is also important is capacity building of national programs. We've been actively engaged in training of national programs by hosting training courses annually, and 11 courses have been done, training over 200 researchers in the area of stem rust research, and also standardizing the note-taking and evaluation of germplasm, trying to network with global researchers at the phenotyping platform in Kenya. And this networking has resulted in in-house capacity building, wherein several staff have actually been trained in different universities and advanced research institutes with different fellowship programs. As you could see, 6 PhD and 12 master students have been trained. For example, a couple of Borlaug fellow programs, award programs, and also Borlaug Leap and BHAT fellowships were awarded to several of the students, including Women in Triticum Award. That facilitated in-house capacity building of researchers to conduct better breeding and better research in target environments. And knowing of the success of these phenotyping platforms with the DRRW, DGGW projects, multiple other projects associated with different research activities have also been conducted at the stem rust phenotyping platforms. And for these efforts that Kenya has put in terms of facilitating global testing of international wheat germplasm, varietal release, and also in terms of capacity building, were awarded the prestigious 2015 Gene Stewardship Award. In Australia, despite all the successes, the challenges are still imminent in these regions. One thing is of the fact that UG99 has been continuously evolving, overcoming several of those race-specific genes leading to boom and bust cycles. As you would see, this is a popular variety Robin, released in 2009, which broke down with the TMP virulence within the UG99 lineage, and also with the Digaloo race group in 2014. And similarly, widespread migration of stem rust races, not only just the UG99, but also more complex Digaloo and the Sicily races belonging to the clade 3 and clade 4, which are now dominating over the UG99 races in East Africa. Another bigger challenge compounding the stem rust problem is the yellow rust races, the spread of aggressive races of the PST S1 and S2 lineage alongside warrior and other races, which are quite adapted to warmer temperatures. I wouldn't want to sort of go back into detail of the migration of these races. The most infamous migration of yellow rust all the way from Europe into Australia dates back to 1979, and also early 1900s from Europe into North America. However, significant transboundary migration of these races have been reported from the northern Himalayan regions all the way into Europe as early as 2011, reflecting the more complex PSTS1, S2, as well as the warrior race group. And the migration of races from Central Asia all the way to East Africa in 2016, the PSTS11 races, and similarly, the migration of PSTS13 races all the way from Europe into Argentina, causing severe outbreaks. The bigger challenge with such kind of diversity in terms of yellow rust race groups becomes a challenge for breeding programs with diverse evolving migrating pathogen populations. And expanded disease phenotyping network, as I mentioned earlier, established for various biotic stresses will enable breeding for multiple disease resistance that is effective against broader geographies and reproduction environments. This phenotyping network opens opportunities to increase coordination in global phenotyping network, thereby avoiding duplications as well as building on the efficiency and capacities for research in close collaboration with national programs. And these phenotyping hubs will play a vital role in capacity building of national programs through training and student exchange programs. So in summary, these phenotyping platforms have played a vital role in not only mitigating the threat of transboundary diseases, but also in terms of combating multiple diseases and also to breed effectively against multiple biotic and abiotic stresses that are prevalent in different geographies. And with that, I would like to thank all the funders for their generous support and also partners with very different projects that have been implemented at the phenotyping platform. And thank you all for your attention. And at the same time, stay safe in these COVID times. Thank you.